and it's after the fact.
Thanks.
All right, so we're on air now. So from the magic of the internet, so it's 4 a.m. here in Tucson. It's 9 p.m. in Victoria, Australia, but we are carrying out a micro seminar for whoever is going to watch either now or in a few hours when everybody else wakes up. But the important part is that we're sharing some cool science and some cool microbiology. So today's seminar speaker is going to be Suzanne Ford. Suzanne just finished her PhD, I think at the end of last year with Kayla King in Oxford, and now has moved, I think around halfway around the world, all the way around the world, to Victoria, Australia at Monash University with Beth McGrath, I think working maybe on mosquitoes in Slovakia. Other than that, like I said, I'm waking up, but this is going to be Suzanne's written some cool papers, and it's going to be an interesting seminar, I think. So without further ado, please take it away, Suzanne.
Okay, I'm going to try and do screen share. Okay, is that right? Yeah, I'm going to say that's okay. Okay, so what I'm going to talk to you guys today is some of the work that I did in my PhD. So the title of the talk is How Coevolutionary Interactions Between Microbes Shape Pathogen Virulence. So infectious diseases come in a variety of shapes and forms. So for example, on the left we have really cool, the infamous zombie ant fungus. This is a parasitic fungus that infects these carpenter ants, and it changes their behavior so that they climb up to the top of plants and they clamp on with their jaws so that they hang, and then the fungus basically grows out of the head and drops onto its next unsuspecting victim. In the middle we have another really cool example. These are parasitoid wasps. They're injecting their eggs into these aphids. And basically the eggs grow within the aphid, and the aphid dies and becomes a mummy, where the wasp then emerges, and again the cycle continues. And on the right we have another example. This is just a classic pathogenic fungus. It's a chytrid fungus. It causes chytridiomycosis in a lot of different amphibian species, and it's actually threatening quite a lot of amphibian species with extinction at the moment. So a lot of what we know about pathogen adaptation and how it infects the host is obviously based on a lot of studies that focus primarily on host and pathogen interactions. What's interesting at the moment, though, is that we're beginning to find more and more that microbial organisms that live within and on the host can actually play a really large role in defending the host from pathogenic infection. And these microbial symbionts are often called defensive symbionts. A lot of really cool examples of these across lots of different hosts. One example is on the left here. You can see this is the hoopie bird. In their preen secretions, they have a bacterium called Enterococcus faecalis that produces antimicrobials. And this protects not only their feathers, but also they put it on their eggs, so their offspring, against pathogenic infection. In the middle, we have the aphids again. And these guys, if they contain a bacterium called Hamiltonella defensa, then and actually in turn, it's not actually the bacterium that protects them against their parasitoid wasps. It's actually a phage, which is a virus that lives within this bacterium that encodes toxins that basically prevents the parasitoid wasp's eggs from developing within the aphid. So they're protected in that way. And then on the right, we have a cool example of the cacao plant. These plants can be infected by a large range of different pathogenic fungi, and actually it's been found that there's quite a lot of mutualistic fungi that compete for host resources but aren't harmful themselves, and so actually protect these plants from the pathogenic fungi. The really interesting aspect of all of these defensive symbionts is a key theme. So most of these symbionts actually act to protect their host, by reducing pathogen fitness. So this is this is an alternative strategy, say, to just protecting the host by making it more tolerant of the pathogen. It actually makes it just more resistant. So basically it reduces the pathogen infection. And so this has really important implications for pathogen, what we think the pathogen might do, because obviously reducing pathogen fitness is applying a really strong selection pressure to the pathogen. And so it's likely to evolve resistance to the defensive symbiont. Despite this fact, not a lot is actually known about how pathogens evolve in response to defensive symbionts within their hosts. And so this raises a lot of questions for me, what I'm interested in. Firstly, of course, how do defensive symbionts interact with pathogens over evolutionary time? So, for example, does the pathogen evolve to become resistant to the defensive symbiont? And then, of course, what does this do in turn for the defensive symbiont? Does it select for that to become counter-resistant? And then, of course, do you get coevolutionary interactions and things like that? And then the second question that I'm interested in is what kind of effects these interactions may have on the evolution of the disease itself. So, for example, if it does evolve resistance to the defensive symbiont, does the pathogen then actually become, does this have implications for how it affects the host? So does it change how it infects it? Does it become more or less harmful? And for me, these questions are becoming more and more important to answer, given that defensive symbionts are becoming attractive candidates for disease control. So, for example, already at the moment, something that I'm working on now in Melbourne is the use of the Wolbachia bacteria in mosquitoes to prevent them from transmitting certain diseases. So, for example, dengue, but also Zika and malaria and other diseases that are vector-borne. And of course, in the middle, we have the example of the cacao plant again. So defensive symbionts can play, are likely to be able to play a really important role in the protection of agricultural crops. And in the case on the right, which I mentioned earlier, in the chytrid fungus of amphibians, defensive symbionts have actually been found on the skin surface of a number of amphibians that could play a really key role in actually protecting amphibians from this fungus. And so there's another example there where they could also be used for species conservation purposes. So in my talk today, I'm going to split it up into 3 sections. The first part is basically introducing the model system that we used to answer the questions. The second part is to focus on the first question, the evolution of defensive symbiont and pathogen interactions. And the final part is basically focusing on the evolution of pathogen virulence. So for part 1, as you can probably tell from this picture, we used the model organism Caenorhabditis elegans. This is basically a cute little worm. It's like 1 millimeter in length, and it's a really useful system for us because for a number of reasons, really. Firstly, it ingests bacteria as a food source, so it's quite easy to get a natural infection. And it's been found that a lot of different bacteria can infect it naturally, usually via the gut. Another really useful thing is that it has a really short generation time. So it has generation time of 4 days. So it's really good for experimental evolution, and it's quite manipulatable in the lab. You can also freeze it down, which is super useful. Another really key thing for bacterial studies or any kind of microbial study is that you can apply bleach solution to a population of worms and it kills pretty much everything apart from the eggs, which the surface protects them from the bleach solution. So this means that you can basically create a sterile environment and a germ-free worm. And then you basically you can control exactly what gets into their gut by what you feed to it on the plate, on your Petri dish. So this is pretty cool. And the pathogen that we used with this C. elegans system is Staphylococcus aureus. This is obviously widely known as an opportunistic pathogen of a lot of different animal hosts. And within C. elegans, it's known to infect via the gut. And the defensive symbiont that we decided to use is Enterococcus faecalis. If you remember, this is the bacterium that I mentioned at the start that the hoopie bird secretes in its preen gland, from its preen glands. And I will show you my first graph. So this is basically the percentage of hosts that are dead after 24 hours, and it's our measure of virulence in this model system. And you can see here that basically the Enterococcus faecalis is not very virulent at all after 24 hours, whilst the pathogen, Staphylococcus aureus, can kill all the way up to 30% of the hosts after 24 hours. And so it's significantly more virulent. And the interesting thing here that you can see is that under co-colonization, the defensive symbiont, so the Enterococcus, is able to bring down the virulence of the pathogen significantly. So this is really cool. And this is why we consider the Enterococcus faecalis a defensive symbiont in this system. So the next graph, we basically looked at the infection load of the pathogen in the absence and the presence of this Enterococcus faecalis. And what you can see here, this is basically the colony forming units per host. So this is our infection load of the Staphylococcus aureus, and you can see that it's significantly reduced in the presence of the Enterococcus faecalis. And from previous studies, we've actually found that Enterococcus faecalis produces superoxides, and these are oxygen-free radicals, and these are in part responsible for this reduction in Staphylococcus fitness, and it acts to slow growth rate. So that's pretty cool. What's also really interesting in this system is that if you look at the infection load of the Enterococcus on its own and in the presence of the pathogen, you actually see that there's a fitness benefit in the presence of the pathogen. So this is really interesting. It kind of suggests that the Enterococcus is actually gaining some sort of exploitative fitness benefit. So it's exploiting the Staphylococcus in some way. And so in this way, the system's quite host parasite-like, where it's antagonistic, where one benefits at the expense of the other. So this is the system that we use, and this leads me nicely onto part 2. So this is where I focus on the evolution of defensive symbiont and pathogen interactions. So as I mentioned earlier, a lot of studies on pathogen evolution focused on the pairwise interactions of a host and its pathogen. And a lot of these studies have focused on how they can co-evolve together. So coevolution is basically, in this case, say you've got a host and it evolves to become resistant to a particular pathogen like this, then coevolution would be the reciprocal adaptation of the pathogen in response. So basically the pathogen would evolve in turn to infect this new host. And this kind of cycle would go on and on and on. And this is really cool. What's interesting to me is when you add defensive symbiont into the mix, if that's doing, you know, quite a large proportion of the defending of the host, then I kind of wonder what would happen to do with the coevolution. So it's been predicted theoretically that in this case, coevolution would actually occur between the pathogen and the defensive symbiont. So this is really interesting, and it's not really known if this is actually happening yet. So there's 2 things that I need to outline at the moment. So just as studies have found between a host and a pathogen, in order for us to test whether coevolution is happening between a defensive symbiont and a pathogen, we need to look for signals of coevolution. And studies between hosts and pathogens have found that there are 2 main forms of coevolution, and so 2 types of signal that you need to look for. So these come in 2 forms, yeah. So the first one is arms race dynamics. This is basically where selection favors generally beneficial traits. So say you've got a trait that is beneficial and increases your ability to infect a wide range of hosts, then it's going to spread to fixation. And likewise, if a host has a trait that is generally beneficial and so allows them to resist a wide range of pathogens, then again, that would spread to fixation. And so this model is basically based upon these kinds of interactions. And so you see recurrent selective sweeps like this, and they tend to result in escalating traits. It's best described with an analogy. So say you've got this host, it's pretty happy because it's not being infected. If you have this pathogen, say it uses a sword to infect this host, then naturally selection would favor the host to evolve a shield to resist this pathogen. Then a directional way of getting around this system would be for the pathogen to evolve a bigger sword, and so then it can better it can better infect the host. In turn, this would select for the host to evolve a bigger shield, which again, a bigger sword, a bigger shield, and this goes on and on and on. And so you can see that this is a generally beneficial trait that escalates through time. And so the second form of coevolution is called fluctuating selection dynamics. And this is different because it's based on more specific interactions between hosts and their pathogens. And it comes about by the fact that if you have a pathogen that can only infect certain genotypes of hosts, then it pays the pathogen to adapt to the most common host genotype in the population. And so as a result of that, it actually pays the host to be the rarest genotype in the population. As a result of this, you get negative frequency dependent selection. And so these genotypes sort of fluctuate through time. Again, this is best, for me, visualized with an analogy. So say you've got this sword and this shield. In fluctuating selection dynamics, a way for the pathogen to get around this would basically be to alter its strategy. So in this case, say it uses chemical warfare. And this would then select, obviously, this chemical warfare is a good adaptation in this case because the shield that was evolved against this sword is no longer a relevant form of defense. And so the parasite that evolves the chemical warfare is able to infect the host really well. In this case, the host is then selected to evolve a gas mask, say, and that is a relevant form of defense, and so it's resistant. In the case of fluctuating selection, this could select for the recurrence of this sword strategy, which again, here the gas mask would not be a relevant strategy against. So it's selected for, and this in turn again selects for the shield, and then you can have a recurrence of the chemical warfare. And so you can see how this would go on and on and on. And actually, rather than an escalating directional trait that's generally, increasing infectivity or resistance range, this is actually just alternative strategies cycling through time. And so what's important about these two different forms of coevolution is that in the first, you would expect to see quite rapid evolution as genotypes spread to fixation. And at any given moment in time, if you took a snapshot, the genetic diversity wouldn't necessarily be very high. And then in the fluctuating selection case, you might not necessarily see any rapid directional change, but at any given moment, you might take a snapshot and you may expect to see more diversity at a given moment. So these two different forms of coevolution might alter how you say a host or a pathogen evolves, and so it's quite important to know which one's occurring. So back to the case of the defensive symbiont, as I mentioned earlier, there's not really any direct evidence of coevolution between a pathogen and a defensive symbiont, and it's mostly been studied with the host. And what has been found is indirect evidence from nature. So in each of these cases, the bumblebee, the aphid, and the ant here, it's actually been found that their defensive symbiont is responsible for the specificity in their interactions with pathogens. So it's basically the genotype of their defensive symbiont is responsible for determining the resistance to a pathogen, and this is instead of the host genotype. So this kind of data suggests to me that coevolution between a pathogen and defensive symbiont is very likely. It kind of suggests that it is actually the case. But of course, as I said, there's no actual direct evidence. So this is kind of what I wanted to do in my study, and I will tell you how. So the most direct way of getting evidence of coevolution is via time shift experiments. Again, this has been done between pathogens and hosts, but not between pathogens and defensive symbionts that I'm aware of. What basically happens here is that you take a pathogen from a certain time point and you infect it into a host that it has seen from its past, from its present, and also from its future. So as you can tell from this, it's kind of, it's not really an experiment that can be done in most systems because you need to have access to hosts and pathogen populations throughout evolutionary history. So it's a little tricky. But what you take from these time shift experiments is basically you look for signals of adaptation across time. And these signals of adaptation across time will be different depending on the type of coevolution that's happening. So for arms race dynamics, you can see here that if you took a pathogen from a mid-time point, so this pathogen with this mid-sized sword here, and you competed it against its host, or you infected it against its host from the past, the present, and the future, you can basically see that it's going to do increasingly better against hosts from the distant past because they're less and less protected against that particular sword. Whilst if you competed it, or infected it, sorry, against hosts from the future, then it's going to do worse and worse and worse because these defense mechanisms are getting more and more relevant. So this is the kind of pattern that you would see from a time shift that where the host and pathogen would be evolving via arms race dynamics. And then for fluctuating selection dynamics, you can see that these signals of adaptation are different and they're more cyclical, and this is obviously just based upon the matching or mismatching of that particular strategy against the enemies, and you can just see that it would cycle through time depending on what strategy is being used at a particular time. So the final thing to note about these time shifts before I actually show you about what I did is the fact that the size of the time shift matters. Now, basically, you can see from this small window, if you were to go and test a pathogen, say against the host, or in our case, a defensive symbiont from the past, the present, or the future, it matters how big a window you look at. So if you took a small window like this, then obviously you can't really see any difference between the arms race and the fluctuating selection dynamics. They basically look the same. But if you took a bigger window, so you went further back into the past and further forward into the future, then you begin to be able to see more differentiation between the two. So you can actually see that there's fluctuation on the one on the right. And so this is particularly important for this kind of study. Okay, so now on to what I actually did. To measure signals of coevolution, I basically performed an experimental evolution experiment with my two bacteria inside of the worm. So I have two main treatments. I have a coevolution treatment, and in these, basically the pink represents the pathogen and the blue represents the defensive symbiont. In the coevolution treatment, I have allowed them to co-evolve effectively, and in the single evolution treatment, I keep them completely separate. And I have 5 replicate lines of each of these. And for each generation, I exposed them to one stock of C. elegans worms. This worm, I must stress, is not evolving. This is just about bacteria. So every generation and every line is being exposed to the same population of worms. So each generation, I expose the worms to the bacteria for 24 hours. After this time, I select 10 worms and I wash their external surfaces, so there's surface bacteria is all removed. I crush them and I plate out the bacteria that's on the inside of the worm onto selective media. I then choose 10 colonies from each species and I grow them up overnight, and I can do the whole thing again. So it's basically, that is one single generation, and I do it 10 times. For the time shift, I basically looked at the biggest window that I logistically could do, and I focused it more on the latter half of the experiment, so generations 4 to 10, as opposed to, say, 1 to 7 or something, because this is all happening de novo mutation. So each bacteria from each line started from one ancestral clone. So I wanted to allow time for the evolution to sort of kick in and any coevolutionary interactions to be underway by the time I had a look. And so for my time shift, I basically chose, say, this one, the pathogen from generation 7, so that the generation 7 was my present, everything preceding it was past, and everything after that was future. I again exposed them to C. elegans for 24 hours and measured their infection loads. So this is basically the infection load of the pathogen from passage generation 7. This is the colony forming units per host, and you can see how it would, how the fitness of the pathogen changes against the defensive symbiont from generations 4 through to 10. As you can see here, the pathogen is most fit against the defensive symbiont that it's recently seen, and again, it's least fit against the defensive symbiont that it has not yet seen, so the ones in the future. So this is really cool, but what's the most cool point is the fact that if you go further back into the past, then actually the pathogen starts to do really badly again. So this kind of suggests actually that the defensive symbiont between generations 4 and 5 actually changed strategy, and you could even go so far as to say that the defensive symbiont genotypes that were hanging around at generation 4 possibly went away and then came back, full force generation 8, 9, and 10. That's possible. But what is most obvious here is that this kind of dynamic is really evident of fluctuating selection dynamics rather than arms race dynamics. So this is really cool, and I kind of freaked out when I saw this. So I felt like the best thing to do was to do the reciprocal time shift and make sure that this was actually happening in both species, and so we can be really sure of this effect. So I went and did the reciprocal, which is basically the defensive symbiont from passage generation 7, and I competed it against the pathogen from the past and the present and the future. And this is what I saw. So this is the fitness of the defensive symbiont. You can see the same general pattern, that it does most well against the pathogen that it recently saw. However, if you go further into the past, it does less well again, so suggesting these fluctuations. What's kind of different is that we are beginning to see another fluctuation happening around generation 9 onwards. So this is suggesting that the pathogen around generation 9 is actually changing genotypes, so the allele frequencies are changing again. And this is, it's interesting here that this is not, the pattern is not exactly the same as what we saw in the defensive symbiont. So this suggests here that the pathogen is actually leading a new cycle of adaptation. And so what you would predict here is that around generations 9 and 10, the pathogen would possibly be doing significantly better in terms of fitness than Enterococcus. So we thought that might be the case, and so what we did was we checked that, and this is basically a graph that shows the fitness of the pathogen. In the middle, you see that it's the coevolution treatment. So this is basically the Coevolved pathogen and the Coevolved defensive symbiont from generation 10 together. And we've compared it against the ancestral bacteria together and also the singly evolved bacteria from generation from generation 10 together. And you can see here that basically the pathogen from generation 10 and the coevolution treatment is doing significantly better than any of the other two controls. So this does suggest that it's kind of winning, but to know that it's winning, we need to know that the defensive symbiont is kind of losing. So we did the same thing with the defensive symbiont, and this is what we saw. So it does seem to be performing significantly worse than the two controls. So at least at generation 10, it does seem to appear that the pathogen is winning. However, we do have evidence of coevolutionary interactions, so that's really cool. So to complement our phenotypic data, we wanted to know what was actually happening in the genome. And basically what we did was we resequenced the ancestral clones, and we also sequenced the populations from generation 5 and 10 from both coevolution and the single evolution treatments. Okay, so in each of these treatments at these generations, we took 40 clones from each of the 5 replicate lines and we pooled them. And so this basically means that we have a population sample of each replicate line from each treatment, from each species at generation 5 and 10. And what we first are going to look at is basically what happened as a result of coevolution. So obviously with our phenotypic data, we have some really cool evidence of fluctuating selection dynamics. This obviously would tell us that we're expecting to see allele frequencies fluctuate through time. So the first thing we wanted to do was actually see whether these alleles were fluctuating in time. So this is what we looked at, and it's pretty cool. So you can see that the pathogen, this is the mutation frequency in each replicate population from generation, so the ancestral population to generations 5 and generation 10. You can see here that the mutations that arose and spread to like quite medium to high frequency at generation 5, a lot of these actually disappear by generation 10 and are replaced by other mutations. So this is really cool, and it's really consistent with the kind of fluctuating phenotypes that we see. Of course, there's a couple of replicate populations there that seem to have some mutations that appear to be sweeping to fixation, which is more evident of, say, arms race dynamics. There's a number of unknowns with this particular data, so it's not we're not sure whether this is whether it's possible that you can have directional selection acting on some aspects of the genome, whilst you can have fluctuating selection acting on other parts. So this is possible. It's also possible that we miss a fluctuation, so say between generation 5 and 10, where we're not measuring the allele frequencies. It's possible that that mutation, went down and then back up again. So we can't really conclude from that whether it is actually arms race or fluctuating, but on the whole, we have some pretty cool evidence that's consistent with our phenotypic data of fluctuating selection dynamics. So that's pretty neat. We did the same thing with the Enterococcus, so the defensive symbiont. And you can see here that you get, again, the same kind of fluctuations, albeit to a lesser degree. So interestingly, we have fewer mutations overall, and they're also spreading to much lower frequencies. And so this is kind of consistent with the fact that our Enterococcus doesn't seem to be doing quite so well in the coevolutionary arms race. It's maybe kind of hinting at the fact that the defensive symbiont might have a lower evolutionary potential. It could be to do with the fact that it's got a lower growth rate. So it's possible that that's why that's occurring. So the next thing that we wanted to look at was pathogen divergence. So obviously it's predicted that coevolutionary interactions are, drive rapid evolution, and we wanted to see whether this was the case between pathogen and defensive symbiont. And so what we did was we focused on the pathogen SNPs and other types of mutations that we saw in our pathogen populations. And we basically created a Euclidean genetic distance tree, and this is basically the square root of the pairwise distances between the ancestor, the single evolved populations, and the Coevolved populations from generation 5 and 10. So here the black dot is the ancestral population. All of the nodes in the grey are the singly evolved populations. The lettering, the numbers next to them indicate whether they're from line 1, 2, 3, 4, or 5, and whether they're from generation 5 or 10. And the pink nodes are the Coevolved populations. Just as an eyeball, you can see that the branch lengths are much longer for the Coevolved populations. So this sort of suggests that the coevolutionary interactions do actually drive greater pathogen divergence. But of course, we wanted to put some actual statistics on that. So we pulled out the pairwise genetic distances from the matrices, from the matrix, and performed statistical analyses on this. So just to break it down, we first looked at the level of pathogen divergence from the ancestor. So we did this by basically comparing the pairwise distances of the Coevolved treatment to the ancestor, comparing it to the distances of the singly evolved populations to the ancestor. And we did this just for generation 10. And we do find that basically the Coevolved populations are significantly more diverged from the ancestor than the singly evolved populations, and you can obviously see that quite nicely from this image. The next thing that we looked at was basically the diversity in evolutionary trajectories. So this is basically answering the question of whether coevolution with a defensive symbiont favors more diverse evolution, and we basically approached that by looking at the pairwise distances between the replicate lines within the same treatment. And again, we do see that the coevolution with the defensive symbiont favoured more diverse evolutionary trajectories. So you can see that the branch lengths here between all of the replicate lines of the Coevolved treatment are much longer, and they're much further apart than the replicate lines of the singly evolved populations. So that's really cool. And then the final thing that we looked at in terms of the genes was basically the rate of genetic turnover. So of course, a specific prediction with the fluctuating selection dynamics is basically the rate of, Okay, sorry about that. Yeah, so basically we looked at the rates of genetic turnover. A specific prediction from fluctuating selection dynamics is that you would expect to see a higher rate of genetic turnover across time, as these strategies are supposed to be fluctuating through time. So we basically compared this, the mutations that were present at generation 5 and generation 10 between the coevolution treatment and the single evolution treatment. And again, this is, we do find evidence of significantly more turnover in the Coevolved treatments, the treatment compared to the singly evolved treatment. So we do have evidence of greater genetic turnover, so that's really, really cool. So to conclude part 1, we found that the defensive symbionts can co-evolve with the pathogens, and in our case, this occurred by fluctuating selection dynamics. And as a result of this, we found that coevolution drove pathogen diversity, which of course has important implications for how we understand how pathogens change over time. So my next question was whether basically these coevolutionary interactions have important implications for how a pathogen interacts with its host, which leads me on to my final part of the talk, the evolution of pathogen virulence. So pathogen virulence is defined as the infection-induced decrease in host fitness. And so it's basically the harm that it causes to the host. And it's assumed to necessarily be a consequence of pathogen replication. So just as it uses host resources, it causes harm to the host. Importantly, there's not really been much literature surrounding how defensive symbionts could affect how pathogens evolve their virulence towards the host. But there has been some literature that has focused on how pathogens interact with other pathogens and how their microbial mechanisms of interaction can shape pathogen virulence. And so I looked at these papers and I thought they were really interesting, and how these microbial interactions are actually similar to what goes on between a defensive symbiont and a pathogen. And so I thought that a lot of this literature could actually probably be applied in the case of the defensive symbiont. And so I made two predictions. The first one is basically linking the resistance of a pathogen to a symbiont and its virulence. And so basically the first prediction is just, if there's a positive correlation between the resistance to a symbiont and the pathogen's virulence, then you will necessarily get selection for increased virulence. Selection for resistance will necessarily increase the pathogen's virulence. And then the second prediction is just simply a trade-off. So if the mechanism of the pathogen's resistance to a symbiont trades off against its virulence, then you will get selection for a reduced virulence. So examples of these cases make it a bit more clear to explain. So in the first case, where there's a positive correlation, this might kind of come about, say, where you have a pathogen that might be able to resist a symbiont, say, by growing faster. Then this growing faster would necessarily result in a more virulent pathogen. In the case on the right, where there's a trade-off, this may come about, say, where if a defensive symbiont produces antimicrobials to harm or kill the pathogen could evolve to resist those antibiotics, but it might come at cost to growth rates. So their growth rate might be slower, and so this might result in reduced virulence. And so each of these cases is equally likely, and they might differ in different systems, but these are the two predictions that I propose. And so what I did was basically use the same evolution experiment that I did before, but I focused on the two pathogen populations from generations 10, and I basically compared their virulence from the coevolution and the single evolution treatment. So here, this is the percentage of hosts that are dead after 24 hours. And this is the ancestral population of the pathogen, the co-evolved population, and the singly evolved population. And you can see here that basically the co-evolved population of the pathogen is significantly less virulent than both the ancestor and the singly evolved controls. So basically, yes, we do find that the defensive symbiont and the coevolution with the defensive symbiont did actually significantly affect pathogen virulence evolution. Not only that, but it actually drove it to be less virulent. So this was really interesting to me. And of course, I needed to know why. So I started exploring. The first thing that I naturally looked at was the pathogen growth rate. And you can see here that basically the coevolution treatment and the pathogen that, so the pathogen from the coevolution treatment that was significantly less virulent is also significantly less, grows significantly slower. And you can see that there's a correlation here between the growth rate and the virulence. So it's kind of interesting. It seems that the pathogen has adapted to grow slower. So this is kind of interesting because it seems a little counterintuitive. So I wanted to double check that this was indeed actually adaptive. So I computed it. I computed the pathogen populations from the single evolution, the coevolution, and the ancestral population, all against a common defensive symbiont to see how well they actually fare. And you can see here that basically the fitness of the pathogen is actually significantly higher in the coevolved populations than both the ancestral and the single evolution controls. So this is really interesting. Despite the cost in growth rate, it is adaptive. It does seem adaptive. So this kind of suggests my trade-off model. So this is basically where selection for resistance to a symbiont traded off against its virulence. So in order to kind of work out what mechanism was going on, I delved into the literature and looked at what kind of mechanisms might cause this trade-off. And then we tested a number of different things. A lot of them didn't work, unfortunately, but we managed to hit on one thing that seemed pretty promising. So there is a lot of literature in the field of pathogen virulence evolution, and some of it focuses on Siderophore proteins. So Siderophores are proteins that are released or produced by a bacterium into the environment. They bind iron, and the iron can then be taken up and used, and so it's really critical for bacterial's growth rate. And so it necessarily correlates, the production of Siderophores necessarily correlates with growth rate, and thus in turn it correlates with their virulence. So this is a really interesting strategy which could be linking our growth rate and our virulence. What interested me most about this particular trait is that it's a social trait as well. So because it's produced into the environment, it can be taken up by neighboring microbes. So this is really cool. There's been a lot of theory about the production of Siderophores like these and other public goods. So this is called a public good. It's been predicted that when the neighboring microbes that basically steal these public goods, when they're not related to the bacterium that is producing the public goods, then that bacterium is selected to produce fewer of them because it's basically being exploited. And so this would result in a lower growth rate and so a lower virulence of that bacterium that's producing the Siderophores. And so this is what we see. We see, a lower growth rate and lower virulence in a social context. So I was beginning to think, well, this might be relevant in our case. And not only that, but of course, this neighboring microbe actually harms the growth of the producing bacterium. So what more, incentive is there to slow the growth of something that's killing you? So this is all, great, but is it actually happening? So I wanted to test basically the production of Siderophores from my pathogen to see whether the defensive symbiont was in any way exploiting it like that, and see if this was actually at play. So what I did was I basically measured the Siderophore production. I used a colorimetric assay. I measured the production in the ancestral population, the Coevolved population, and the singly evolved populations of the pathogen. And you can see here that the Coevolved populations of the pathogen are producing significantly less Siderophore than both of their controls. So this seems really, really promising, and I was really excited by this. So, regardless, the coevolution with the defensive symbiont does seem to have affected Siderophore production. So the next thing that I wanted to know was, does this actually have any effect on the defensive symbiont? So what I did was basically I grew the defensive symbiont, the Enterococcus faecalis, in vitro with increasing concentrations of Siderophore, and I used the same kind of range of Siderophores that Siderophore concentration that I knew that Staphylococcus was producing. And you can see here that with increasing Siderophores, you can see a really strong effect on the benefit to Enterococcus. So it's growing faster and faster with more Siderophores in the environment. So it's pretty clear here that Enterococcus is able to exploit Siderophores in the environment. And so with this information and the information from the previous slide, it does seem to make sense. So we should predict that the ancestral population and the singly evolved population of the pathogens would probably benefit the growth of Enterococcus by producing more Siderophores than the Coevolved population, because that produces significantly fewer. So I went on to explicitly test this, and what I'm showing you here is an in vitro experiment where we have the fitness of the Enterococcus defensive symbiont, and this black line here represents the fitness of the Enterococcus without the pathogen. And you can see that with the pathogen, so you see a significant fitness benefit with the ancestral and the singly evolved pathogens, whereas in the center you don't see a significant benefit in fitness with the Coevolved population. So this makes sense. The Coevolved pathogen is producing fewer Siderophores, and so this fitness benefit for the defensive symbiont is completely removed. So this is really, really cool. But I wanted to really just make sure that the production of the Siderophores is what is responsible for this. And there's one really neat way that you can deal with that. So basically, if you add in exogenous iron, so like lots of iron, it's thought to down-regulate the expression of Siderophore because it's not needed as much. And so you might be able to eliminate that, that difference between these populations. And so this is what I did. You can see on the left here, this is basically the graph that I just showed you, the differences in defensive symbiont fitness with the three populations of the pathogen, and you can see the significant differences there. And on the right, you can see the fitnesses of the defensive symbiont in the same pathogen populations, but with added iron. And so you can see that the differences are removed. And so it kind of suggests that this difference in symbiont fitness is to do with the evolution of Siderophore production. So that's super cool. I was really happy with that. But this is all well and good, but what we really want to know is actually whether the changes in Siderophore production by the pathogen in response to the defensive symbiont is actually responsible for the changes in virulence that we saw. So I correlated that, and it turns out, yes, it is. It's pretty cool. So you can see here that the Siderophore production along the bottom axis and the virulence of the pathogen along the y-axis, you can see that it increases. So the Siderophore production seems to explain pretty well the virulence that we see in the pathogen. What's interesting here is actually this point here. So at the start, we didn't know what was going on until we got our genomic data back, which is quite a bit later than when we got this data. And then by looking in the SNPs, we actually found two SNPs in this particular population here in Siderophore iron reductases. And so actually it's not, in this case, it's not a reduction in Siderophore, but rather an increase in Siderophore iron processing. So it produces the same amount, but it seems to take the iron from the Siderophore faster. So it's kind of an interesting case of parallel evolution of alternative strategies around the same kind of thing. So that's really cool. So we have an explanation for that appearing outlier, which is really nice. So yeah, that's about that. And so to conclude my final point, final part, we found that defensive symbionts were able to drive virulence evolution in the pathogen. And in our case, we found that the reduced virulence was a byproduct of the adaptation to the defensive symbiont. So this kind of data supports the trade-off model that I presented at the start of this section. But of course, the other model, I don't think, that could very much happen in other systems where different mechanisms are happening. So I think that the direction of virulence evolution, it could go one way or the other, and that would be very much likely dependent on the mechanism of interaction between the symbiont and the pathogen in each case. And so following that, I feel like this data has quite important implications for disease control, not only because we're able to show that the symbionts can change how a pathogen interacts with its host, but it also could inform which defensive symbionts you're likely to choose. So if you knew that a defensive symbiont interacted with a pathogen in a way that would cause it to become more virulent, that would be pretty bad. You'd want to avoid it. If you could identify that it was interacting with a pathogen in a way that actually caused it to trade off resistance on virulence, then that would be pretty cool. I'd be pretty happy with that. So that's my talk, and I'd like to thank my supervisor Kaylee King for all of her help and guidance throughout the project. And I'd also like to thank other people that were in my lab at the time, my co-supervisor Stu West, and also the collaborators that I had at University of Liverpool that helped with some of the genomic data. I'd also like to thank University of Oxford, St John's College and EPSRC for the funding that enabled me to do this. So thank you very much for listening. I hope that wasn't too long. I think it was a little bit, but never mind.
That was awesome. That was awesome. Great job.
Bye.
You missed my my computer completely melted down during the middle of it too. So my frantic scrambling was it all worked out though. One question.
Okay.
One question. So in terms of, so you have the genomic data, you have the SNPs and stuff from this. Do you see a lot of parallel evolution aside from the Siderophores within the populations, or is it like can you see cool evolutionary dynamics and parallel evolution when you sequence across these populations, or are the mutations kind of all over the place?
Mutations are all like within things that can be linked to the Siderophore pathway, and they're kind of similar, but there is some like differences. So we have ones on like ABC transporters and things like that. So in terms of the pathway, not necessarily Siderophores themselves, but then we also have ones that are actually involved in like the Siderophore itself. So it's kind of like different aspects of the same pathway. And as obviously as that last example where, you know, we thought what's going on here, it's not reduced Siderophore production, it's something else. It's like the processing of it. So yeah, we kind of have parallel but alternative. And I never got the same mutation twice either. So it's quite cool. Yeah.
Cool. Yeah, I'm thinking a lot more recently too. So I do a lot of bacteriocin work, and a lot of the bacteriocins actually target these Siderophore receptors too. So it seems like there could be a lot of interesting, and who kills who, and then how the coevolutionary interaction is affected by that. So.
Yeah, that's so cool. Oh, I just realized that I'm just showing you yourself. I don't know if I should put that. I don't know how to put that back. Anyway, that's fine. Oh, here we go. I'm sorry, carry on.
Yeah, no, so I'm thinking that it, depending on whichever, like you can select it for which strains kill each other, and then if there's a trade-off with Siderophore utilization, you might get completely different trajectories with how the evolve. So it'd be pretty cool.
Nice.
Look around. So you did have viewers. Don't worry. There were people that were awake watching. So.
Ooh. Am I live now?
What?
Am I live now still?
Yeah, you're still live. I'm gonna end it soon. I'm just waiting to see if anybody has any questions, and then we're good. So thank you very much again. I'm gonna stay on, but I'm just gonna stop the broadcast. But just thank you very much. It was great, and this is I always love getting the evolutionary experimental evolution models out there. So.
Oh, nice. Thank you.